Welcome everybody to our very first Science by the Glass of 2021. Today, I am delighted to welcome Dr. Emily Bailey. She is one of our very own Texas Tech faculty in Abilene at the Health Sciences Center there. She comes to us originally from North Carolina, where she was at Duke, and she is an assistant professor and also the director of student success for our Abilene campus. Now, Emily probably wasn't thinking about this a year and a half when she arrived at Texas Tech studying viruses and the environment, but it turns out that her research is immediately relevant to what's on all of our minds today, which is COVID. So she has agreed to give us a short talk on climate change, COVID, and the environment, and talk a little bit about her own research too. And then she's going to take your questions. So if you have any questions as we go along, feel free to put your questions in the chat at any time. And after Emily's finished, we are going to take those questions live.
So as Dr. Hayhoe said, I am Emily Bailey, and I am with HSC on the Abilene campus. And I've put a little bit back in here about my background. I am from North Carolina. I did my PhD at Chapel Hill, and then I was at Duke for a long time as a postdoc. And I actually did work on zoonotic viruses for a while before we had COVID. And any of you that were working on viruses or zoonotic disease, you know that we were looking at influenza and not coronaviruses to be the next pandemic. So I'm sure that a lot of you are surprised, just like I was, to have this coronavirus coming out of China when we were thinking it was going to be H7N9. Could still be H7N9, we don't know yet, but for now we're dealing with COVID. So I'm sure that many of you have had background on coronavirus. I just stuck this in here so that we could talk about it a little bit before we move forward into the research. Coronaviruses are weird. I think that's a good place to start. They are RNA viruses. And the question is, why do they survive so long in the environment when they are RNA viruses? And I think that's a great question. Something that I face every week is that crazy uncle on Facebook saying, why do we have this virus floating around when we've never had any major viruses before? And I have to keep saying, well, this is called SARS-CoV-2 for a reason. There's a SARS-CoV-1, there's also a MERS, there are also lots of other ones. Coronaviruses are not new, and there are coronaviruses that, impact people all of the time. The ones that we see on a clinical level, those of you that might be medical doctors, we see 229E, OC43, NL63, HKU1. Those are the ones that people typically have. They might be the ones that you have cold symptoms, you have influenza-like illness, but they're not severe. So the issue here is that when a coronavirus is transmitted, there are lots of types of animals that it can transmit from and transmit between. So there are animal coronaviruses in bats, pigs, cattle, all of these different types of animals. They are enveloped. So when we think about viruses in general, a lot of the hardier ones are non-enveloped. And what that means is, in this little image, you can see, let me move my image around. So in this little diagram, you can see that there is an envelope around the coronavirus. And it's contrary to what you would normally think. When a virus has an envelope, that makes it more susceptible to disinfection. It's like it is more like a human particle, more like something that it's recognized by a disinfectant, something like that. And when viruses are non-enveloped, they're much more successful in the environment. And just giving you an idea, when we look at them under the microscope, they are this circular, almost oval shape. They're pleomorphic, and they have a little halo around the outside. So before, when I was saying they transmit among different animal families, different groups, you can see here that the colors are the alpha, beta, gamma, and delta coronaviruses. So in this little diagram, I know it might be hard to see, but you can see, well, maybe you can't see, but If you look a little closer, you can see that the human coronaviruses are mixed right up next to the bat coronaviruses, right up next to the pig coronaviruses. And that is true for the really infectious ones like SARS over here, right next to a bat coronavirus, SARS-CoV-1 right next to a bat coronavirus, as well as 229E, which is that human one that we see every year in people, and TGEV, which is a pig virus that's also very infectious in pigs. So the issue with coronaviruses is that the sequences of these viruses are not really that different among the different groups of animals. And you might think, well, why do we care about that? And the reason is because, well, they are crossing species. They probably cross species all the time, but we can't track it because it happens so frequently. And in this case, we don't know when SARS-CoV-2 crossed because we don't even know what animal it crossed from. So those who are watching the news and reading things frequently, we're still looking for that cross species. A lot of people postulate that it's bats, and maybe some of you are researchers on this and you know more than I do about the search for the early crosses of coronavirus, and maybe that original cross has been located. Another issue with viruses is that when they replicate, they have lots of copy errors, and those errors result in changes to the virus. And so we have variants, and those variants can be different qualities. And by qualities, I mean they might be more transmissible, they might be less infectious. But so far, we've seen a couple different variants, and those variants so far have been more infectious or easily transmissible between people. So in terms of climate, I think back early in March, we thought there's going to be this big climate difference. When the summer comes, coronavirus is going to go away. We're just waiting for the temperature to heat up. Unfortunately, those of us in Texas already knew that it was 60 degrees every day, even in January, and people were still getting sick from coronavirus. And partly that has to do with the way that this virus is transmitted. And partly that has to do with how people react to the coronavirus. Hot weather does take people outside, and there's less evidence of transmission of coronaviruses outside, but there's also no evidence between climate or climate change and the transmission of coronavirus. So I took this from WHO just to put in what their disclaimer is. They have not noted that there is a direct connection between climate and COVID disease, but I think the relationship really is between people and their interaction with animals. If the climate is changing and people interact more frequently with animals, then that is a relationship we have to look at when we're looking at tracking diseases. If any of you have been to China, you might know that the open bird markets, we might call them bird markets, we might call them seafood markets, whatever you want to call them, they are live animal markets, and the live animals are vastly different species. And so when you go into a market and there are 30 species with, waste and wastewater in lots of different places, it's really difficult in terms of biosecurity or biocontainment to think about how bacteria or viruses are being transmitted in that kind of environment. And in China, it's not necessarily that they are looking for food sources or looking for additional access to these types of foods. It's because it's a desire for exotic or specific types of animals. And we don't necessarily have that in the United States in the same way. We're not looking to have a chicken slaughtered for our dinner tonight, but it is a cultural thing in China, whereas people might want to have a live chicken slaughtered for their meal that evening. So this idea of strengthening our health systems and our surveillance of infectious disease is really important if we plan on tracking pandemics globally. Unfortunately, we have not done a good job of tracking pandemics or even monitoring transmission of disease. It's not really an integrated global health system. And it would be very hard to make it one, but it's something that is a a goal that many nations share. I think that the WHO released a report this week about how the response to coronavirus was, substandard, not as good as it should have been. But a lot of that was on their backs, right? Because they didn't have the type of response that they needed to have. They didn't send investigators into China early enough. They just didn't have the type of authority that they needed to have. So one thing I wanted to say about climate is this type of change in coal usage and how different it was in 2020 because of the outbreak. So the coal consumption, the electricity consumption, the gas consumption, all of those things were down last year because of coronavirus. And that is definitely an impact on climate and climate change. Air quality standards and air emissions were down so much last year. I didn't put those figures in here, but huge changes. Staying at home makes a big difference on how much energy we're consuming. I think if I counted the days that I drove my car last year, it would be probably a third, half, maybe a fourth of what I drove the year before. And so if we all think about it that way, it will be very different than how it was previous years and how our consumption could change. And perhaps moving forward, we will reevaluate how we travel, how we spend our time. And here's another view of this. This is in 02 in China, and you can just see the difference between, I can't see the top, 2020, January 2020 and February 2020. There's just a huge difference in close to Beijing. And if you've been to China in the winter, the air quality is very poor, especially during the winter when coal burning is really, really popular and widespread. So another view of this, the change in global daily fossil fuel emissions, huge declines all the way through May. I think some of it did pick up back in the late summer when people started to reopen their economies, but it really just jumped down in March. This is a little blurry, but this little tub, the water level on the tub is representing the amount of carbon dioxide in the atmosphere, and the black line is the end of 2019, and then the green line is the end of 2020 with the pandemic, and the orange line is the end of 2020 without the pandemic. So the pandemic has slowed the flow of CO2 just a little bit, but it's not a huge amount. It's still a giant increase from 2019 to 2020. So yes, we did cut back a whole lot. The whole world cut back a whole lot, which is fantastic. Great. We have decreased our consumption, but we have still made a huge jump from 2019 to 2020. So I'm going to talk a little bit about what my group is doing in research, and I'd love to hear what questions you might have. But I'm primarily working on asymptomatic transmission and trying to figure out what that looks like. Some of that is based on transmission in athletics and what that means on college campuses. And because I'm in Abilene, I've been working with ACU because they have stayed in person this whole time. ACU's campus has about 5,000, maybe closer to 6,000 students. And we're looking at basic prevalence, and they're setting their priorities for campus openings, campus closings on this. And overall, there was very low transmission on campus, except for a big spike after a football game. And after fall break, they did not come back, just to give you a general idea of what happened. They did not come back after fall break. And fall break was canceled. They didn't come back after Thanksgiving. And then fall weekend, homecoming, all of that was virtual, and physical distancing was required. There were no fans at the games, and face masks were required on campus. And if you had COVID, you were required to report that to the university. So in terms of athletics, before you could practice, you had to have a PCR negative. So just a little background on testing. I know this has been in the news, but there are two types of testing that are currently used. One is the antigen test, and then one is the PCR test. The antigen test tests if you have antibodies to the coronavirus, so that means you're actively sick. And the PCR test tests if you have the virus in your nasal passages or saliva or whatever sample you're going to test. And that evidence of the genetic sequence doesn't necessarily mean you're infected, but it does mean that you have evidence of the virus. So the PCR test is very sensitive. That's the test that we're using. And it is definitely slower to get the PCR test than it is to get an antibody test. The antibody test takes about 20 minutes. Whereas the PCR test, if any of you have done lab work, you have to extract and you have to run the PCR. So it can take, depending on who's doing it, anywhere from 1 to 5 hours. So, and how many samples you have. It depends on how many samples you have. And the NCAA this fall required that all in-season teams had a PCR test within 72 hours of a game. And then if you were out of season, you had to be tested. 30% of the team had to be tested every other week. So initially, we tested every student athlete on campus, which ended up being about 450 students. And then every other week, if you were out of season, which was everything except football and soccer, you were tested every other week, 30% randomly selected. And then if you were football or soccer, you were tested every week. So these are the inclusion-exclusion criteria for the study. And most people were tested over and over because they were playing football or soccer. So we collected general data on how many people lived in their household, what kind of work they were doing, and what kind of sport they were playing. In this case, most of the people in the initial screening, it was 354. Most people were not positive in their initial screening, but they may have had COVID before. About 11 people had coronavirus beforehand, whereas most of the people did not. Some of them had symptoms, but what constitutes a symptom early on in the process, almost anything could be a coronavirus symptom. Most of the ones who reported loss of smell were eventually confirmed as a coronavirus positive, and some of the other symptoms have been limited now as we move forward in this pandemic. So ACU has a really nice dashboard that shows you the positives and the negatives. This is from October 13th, so it's a bit dated, but you can see that in October, they had a bit of a spike. This is the dashboard from October. It was very low case numbers from all the way from July through August. And then in October, it hit a bit of a peak. And a lot of the reasons for this are the traveling football team. They ended up traveling to Virginia, and that team in Virginia, either some interaction with that team or the travel on the airplane itself caused this spike that almost the whole football team came back positive after that game. So in the initial screening, you can read the summary here, but one of the issues with testing of COVID is that asymptomatic people are potentially able to transmit coronavirus to others, and the duration of that time between you being able to transmit coronavirus and the time that you test positive is a big difference. So in our initial screening, we found about 5 people to be asymptomatic carriers, which is about normal for back in August. But now I think community transmission is up much higher. So the difference between 6-8 months ago and now is a huge difference. Another thing I stuck in here is this myths about coronavirus. A lot of things that I've heard, a lot of things that people bring up in conversation. First off, this, just like what I've just explained in this study, asymptomatic people can't spread COVID-19. Unfortunately, with this virus, asymptomatic can spread the disease. They might have mild symptoms that they don't even notice. And they can spread it for many days and be not symptomatic that whole time. And then the second one, pets can't spread COVID-19. There's no evidence that pets spread it between themselves and humans, but you can actually give it to your pet, and that would be very unfortunate. But there is no evidence that your pet is going to give it to you. And then all masks provide equal protection. As scientists, we know that's not true. Masks are very different. A surgical mask is not an N95, but you don't really need an N95 to go to the grocery store. So wear the masks that are recommended by CDC, WHO, and think about why you're using it, how you're using it, what is the point of that mask. I get a lot of questions about why masks were not recommended in the beginning of the pandemic and why they are now. And some of that has to do with supply and demand. In the beginning of the pandemic, we weren't able to get supplies because our economy was closed. And so, no, the CDC did not recommend wearing masks. And now masks are freely available. People have made them. People have provided them. Our employers have provided them to us. And so now it is appropriate to wear a mask everywhere you go. So moving forward, there are lots of questions on what it means to be asymptomatic. This is something that my group is investigating, looking at why and how long you can be asymptomatic. And then a question around this is, are people actually shedding the virus when they are positive but asymptomatic? Unfortunately, to really get at that question, you need to be in BSL-3, and during a pandemic, we're not going to get approval from the Biosafety Committee to do to culture SARS-CoV-2. And another question is about this length of PCR positivity. Right now, we know that people who are coronavirus positive then continue to test positive on the PCR for up to 90 days. And that is a really long time to be positive on a PCR. If you've ever had influenza, you don't test positive on the PCR that long. So why do we test that long? And why do some people not test positive that long? Some people only test positive for a month, 2 months, whilst other people are testing all the way out to 90 days. And then the last question is about influenza season and COVID. We actually didn't see a huge spike of influenza in the United States this year, but was that because people were staying inside, staying away from each other, and not really interacting when they had COVID symptoms? It's a good question. But a lot of this stems from not knowing what was going on in China ahead of time and not really being ready for a global pandemic. I'm going to close this, but I have a personal story. One of my good friends is from Wuhan, and she went back to Wuhan to be with her parents in November 2019, and she was sending me WeChats because iMessage doesn't work in China. And she said that, she was going to go back because she knew there was a virus and that her parents needed help. So she goes back to Wuhan in November, and she says, this is really bad. There's going to be this terrible virus. And she's like, why is it not on the national news, the international news? And I said, well, I don't know. We haven't had it on our news yet. And we didn't even see it on our news in the United States until December. And then we didn't see a response until January, February, and then we didn't see a real national response until March. So there are lots of good questions around this, and hopefully you guys have questions. I haven't seen the chat yet, but I'll bring it up.